Thank you, Johannes, for that overly generous and kind introduction. I'm very honored to be here and to be giving this Birnstiel lecture. For personal reasons, I'm trying to cut down on my travel, but I'm old enough to know that this is one of the great research institutes in the world, and so it's very special for me to be here. So here are the obligatory disclosures that you've been wondering about. I wish I could say these were large financial interests. They're small financial interests, but we're supposed to disclose them anyway. Here are the people who've done the work in my laboratory over the years. I'll try to mention some specific individuals as I go along. And frankly, I can't list all the collaborators on one slide who have helped us, but here are some of the key individuals. So what I thought I would do for the benefit of the students is maybe spend about 10 or 15 minutes on some review and then move into some newer things that we're doing. So for those of you who don't know, von Hippel-Lindau disease is a relatively rare hereditary cancer syndrome. It's caused by loss of function mutations in the VHL tumor suppressor gene. This tumor suppressor gene is a classical so-called two-hit tumor suppressor gene, so it's inactivation of both the maternal and the paternal allele that gives rise to cancer. The characteristic tumors that one sees in this disorder are blood vessel tumors called hemangioblastomas of the brain and the retina, clear cell renal carcinomas, the most common form of kidney cancer, and adrenal gland tumors called pheochromocytomas, or sometimes called paragangliomas. And as you would expect from the knowledge that germline VHL mutations predispose to these various cancers, if you now look at, for example, non-hereditary clear cell renal carcinomas, you again see that the VHL gene is frequently mutated, but here the mutations have occurred somatically, in contrast to VHL disease where the first hit has occurred in the germline. Now, students sometimes ask me, why did you even work on VHL disease? How did you know about VHL disease? Why would anyone work on this rare disease? And part of the answer is shown on this slide. So when I was much younger, I actually was a clinical doctor, as Johannes mentioned. And back in the day, and this is in the 80s, one of the things you did as a medical doctor is you memorized long lists like this. Now people just use their smartphone, but in the old days, if the professor said, tell me all the various causes of excess red blood cell production, you were expected to have memorized a list that looked like this. So some of the things are easy to remember. So for example, if you move to high altitude, it kind of makes sense that your body would start making more red blood cells. So I was at the Jungfraujoch 2 days ago, so I'm sure my body was making more red blood cells at 12,000 feet. But also some tumors cause patients to make too many red blood cells, and it always struck me as quite odd that the 3 cancers or tumors seen in VHL disease make this list. Now, kidney cancer is actually pretty common, so maybe that's there by just chance. But cerebellar hemangioblastomas and pheochromocytomas are unusual tumors, and yet they also make the list. So there's some link somehow between VHL and red blood cell production. And then another thing that's known to clinicians is that the tumors seen in VHL disease are very angiogenic, very rich in blood vessels, and that's because these tumors produce lots of VEGF, whereas the red blood cell production is because these tumors occasionally produce erythropoietin. And what VEGF and erythropoietin have in common is that they're normally induced when cells are not getting enough oxygen. So if you're a cell or tissue not getting enough oxygen, this is part of the stress response, is to produce things like VEGF and EPO to try to improve oxygen delivery. Now, we knew from the work of many laboratories around the world that these hypoxia-inducible mRNAs such as VEGF and erythropoietin are under the control of a heterodimeric transcription factor called HIF, or hypoxia-inducible factor. And we knew from the work of Greg Semenza and Frank Bunn and others that the alpha subunit is normally degraded if cells are getting enough oxygen. But if oxygen is scarce, HIF alpha accumulates and dimerizes with HIF beta and turns on these various target genes. And so with this and other clues, we and others went on to show that the VHL tumor suppressor protein is actually part of a ubiquitin ligase complex that in the presence of oxygen targets HIF-alpha for proteasomal degradation, whereas if the VHL protein has been mutated or oxygen levels are low, now HIF-alpha can accumulate and dimerize again with beta activating genes such as VEGF and EPO. Now, this left open. The question, of course, is how does the VHL protein know whether oxygen is or is not present, and hence whether it should or should not destroy HIF-alpha? And it turned out the answer, which we and Peter Radcliffe arrived at simultaneously, was that in the presence of oxygen, HIF-alpha gets hydroxylated on one of two prolyl residues, and this generates the binding site for VHL. And at the time, this was the first example of prolyl hydroxylation being used as an intracellular signal. And Steve McKnight's group and Peter Radcliffe's group, as well as some work done in our laboratory, showed that the enzymes that do the work are the EGLN prolyl hydroxylases, also sometimes called the PHD prolyl hydroxylases. These enzymes have low oxygen affinities, even though they require oxygen. They have low oxygen affinities, and hence they're oxygen sensors. They're very sensitive to changes in oxygen. They also require iron, which explains an old observation, which is if you treat cells in cell culture with iron chelators, you can activate a hypoxic response. And then finally, and interestingly, and this becomes important later in the talk, these enzymes also require a Krebs cycle metabolite called either 2-oxyglutarate or alpha-ketoglutarate, which is then going to be decarboxylated to succinate during the hydroxylation reaction. So it turns out there are three members of this family, EGLN1, EGLN2, and EGLN3, and I give the alternative names here. But multiple lines of evidence suggest that the primary regulator of HIF stability is EGLN1, also called PHD2, and perhaps for that reason you can't make it through embryogenesis without EGLN1, whereas to a first approximation, these are simply spare tires, and you can make it through embryogenesis and make a viable mouse without EGLN2 and EGLN3. So because of this embryonic lethality, Andy Minamishima in the lab did what you would do, which is perhaps make a mouse where you could conditionally inactivate EGLN1 at will using Cre-Lox technology. So here are two mice, both of which are ubiquitously expressing Cre-ER, but this mouse also is homozygous for a floxed EGLN1 allele. And if you give these animals tamoxifen, and I don't know that this projects too well, this paw is slightly too red and these vessels are engorged. And that's because these mice are developing profound levels of polycythemia. So here, blood's been put in a capillary tube and then spun down. So here's the packed red blood cell volume, also called the hematocrit. So a normal hematocrit should be 50% of the column height, whereas if you knock out EGLN1, now 90% of the column is red blood cells. So these animals are making industrial amounts of red blood cells. So that's the mouse genetics, but one thing that trumps mouse genetics is human genetics. So we now know from the work of Frank Lee and Joseph Burkal and others that certain families around the world that produce too many red blood cells on a genetic basis have either hypomorphic homozygous mutations of VHL. I contrast that with VHL disease where you have one wild type allele and one grossly defective allele. These families have two slightly defective VHL alleles. Or they have loss of function mutations in EGLN1, or they have gain of function mutations in HIF2 alpha. So there's very good genetic evidence in mouse and man that this is the oxygen sensing pathway, at least with respect to the control of red blood cell production. And it was very gratifying to see, this is an analysis done by Peter Radcliffe and colleagues, that this pathway is conserved throughout Metazoan evolution. And I understand there are a lot of people who work on model organisms here, so you can probably find your favorite model organism here. But this goes all the way back to Trichoplex. I don't know if we have anyone working on Trichoplex here. But certainly flies, worms, everybody has figured out that you want a HIF, you want a prolyl hydroxylase, and you want VHL. Now, not surprisingly, a lot of these genes were duplicated or triplicated later in evolution and have assumed some new functions as they went along. No. So let's return now to this mouse. So what I didn't tell you is in this mouse, all of the polycythemia is being driven by EPO, erythropoietin, being produced by the kidneys. And that's not unexpected because in the adult mammal, the kidneys are the major source of erythropoietin, but that's not true during fetal life. So during fetal life, the major source is the liver. And then about a day or two after birth, the hepatic EPO locus gets silenced and the kidney takes over. And this has medical and economic implications because, for example, in the United States, there are at least 20 million Americans with chronic renal failure, and on the order of 2 to 4 million of these are anemic due at least partly to a relative inadequacy of renal EPO production. And so Andy Minamishima in the lab wanted to see if the liver could remember, if you will, how to make EPO. And so what he did was he genetically knocked out the different prolyl hydroxylases, eglin 1, 2, or 3, either singly or in every pairwise combination or all three in the liver. And for good measure, for comparison, he knocked out VHL in the liver. And he then measured hepatic EPO mRNA levels and circulating serum EPO. And what he saw was if you knock out all three prolyl hydroxylases, you see a very robust induction of hepatic EPO. Now this is slightly misleading because if you look at this in greater detail, what happens is if you knock out the primary prolyl hydroxylase, you do get a pulse of EPO, but then it comes back down as the two other paralogs kick in. And so you can either get a pulse if you want, or you can get steady state. To get steady state, you knock out all three. So it turns out these prolyl hydroxylases have druggable pockets that even a non-structural biologist like me from the back of the room can see. So this is work from Amgen where they've solved the structure of eglin 1, and they're showing you the region of eglin 1 that accommodates that alpha-ketoglutarate cofactor. And so a number of drug companies have now been developing inhibitors that act as alpha-ketoglutarate competitive molecules and turn these enzymes off. Now, these drugs are orally available. They've gone now, the furthest along has gone into phase 3 trials, but here are some data from a phase 2 trial where patients with chronic renal failure are being given orally a prolyl hydroxylase inhibitor. And on the left, you see that in patients getting this one compound called roxadustat, you see a nice increase in plasma EPO. The patients don't develop tolerance. If you redose them a month later, you can again turn EPO on or off with this orally available agent. And this then translates into improved hemoglobins in these patients with chronic renal failure. Now, I have to tell you, when I was a medical student, I did dream that maybe one day I might touch in some small way a chemical that would eventually become a drug that might help patients. Little did I know that the first patients on Earth taking this drug are actually professional cyclists who have figured out that you can buy these things from Chinese chemical companies, and I guess they're making teas or something, and they're doping themselves with prolyl hydroxylase inhibitors. And I read recently that these drugs are now in the new panel that they're using to screen professional bicyclists. So my day job is to work on cancer, and so this is just to show you that kidney cancer actually is a relatively common cancer in the developed countries in the world. It's usually in the top 10. Now, we know from a variety of studies, however, that even though inactivation of VHL is a critical step in the development of kidney cancer, it's not sufficient. And so, for example, if you look at VHL patients, they're germline VHL heterozygotes, and then what happens over time is some cells in the kidney lose the remaining wild-type allele, but they only become pre-neoplastic renal cysts, and they'll just sit there unless they acquire additional mutations that then convert some of these cysts into actual renal cell carcinomas. And so that says that VHL loss, even if it might be necessary, is not sufficient for the development of these tumors. And so that begged the question, well, once you get to this stage, does VHL even matter anymore? Does HIF even matter anymore? And if it is HIF, which HIF is it? And so over the years, we did a number of conceptually simple sort of necessity and sufficiency types of experiments. And so we showed many years ago that if you restore the function of the VHL protein in a VHL null renal carcinoma cell, they grow happily on a plastic dish, but they lose the ability to form tumors. So that was good. And then to ask whether it was really about HIF, Keiichi Kondo in the lab took these cells and introduced forms of HIF alpha that couldn't be prolyl hydroxylated because the proline sites had been mutated to alanine. And he found that if he did this with HIF2 alpha, these cells could once again form tumors. Conversely, if he eliminated HIF2 alpha, initially with hairpins, but more recently with CRISPR, these cells lose the ability to form tumors. And so this was a little bit unexpected at the time because the well-studied famous HIF was HIF1. HIF1 doesn't do this, so this is something peculiar to HIF2 alpha. So in the mid-90s, and certainly by the year these years, my dream again was, well, some smart person is going to develop a small molecule that will inhibit HIF2 alpha. But to the students, I can't tell you how many things I've been told over the years that couldn't be done until someone showed they could be done. So for example, I remember being told in the 80s, you can't inhibit kinases with small molecules, just forget about it, okay? And in the 90s, I was hearing, you can't drug transcription factors like HIF2 alpha, this is a protein-protein interaction, just go home. All right, well, fortunately, Rick Bruick and Kevin Gardner at UT Southwestern ignored that advice, and so they identified in the HIF2 alpha PAS B domain a potentially druggable pocket. They then did screens in academic labs, and they identified some scaffolds that could bind to this HIF2 PAS B domain, and once bound, induce an allosteric change such that HIF2 alpha wouldn't bind to ARNT. So that was good, but my pharmaceutical colleagues, and I see one in the second row, would tell me that these chemicals were publication grade, but they were not pharmaceutical grade. Okay, these are not drugs yet, and so these needed a lot of med chem. So these were out-licensed to a company called Peloton that did some med chem, and they were kind enough to provide us with a tool compound that is closely related to the compound that has now gone into the clinic. So Chin Frosch in the lab started treating VHL null renal carcinoma cells with this compound, this HIF2 alpha inhibitor. So here she's treating cells at 0.2 to 20 micromolar, and she's measuring the mRNAs for these 6 genes here. And so the first thing she saw was that the message for HIF2 alpha is not affected. That's good because there's no evidence that HIF2 alpha autoregulates itself. And then 4 of the 5 HIF targets are downregulated at high nanomolar concentrations. That's good. And actually, the exception here is quite important because BNIP3, even though it's a HIF target, is a HIF1 specific target. It's not regulated by HIF2. So by this and other criteria, this all looked pretty good. She then did what you and other cancer biologists would do, is she did some efficacy studies. And so here you're looking at nude mice that have orthotopic renal tumors formed by VHL null renal carcinoma cells that are constitutively expressing luciferase, so we can easily monitor the tumor burden. And in the animals treated with vehicle, the tumors keep growing, and that's shown in red. If you treat with the HIF2 alpha inhibitor, the tumors either stop growing or in some cases they regress. Now that's, again, encouraging, but cancer pharmacologists are sometimes appropriately criticized for using models of organ confined disease. So we also obtained from Joan Massaguet a metastatic version of these cells that if you inject them into the tail vein will now form these little pulmonary nodules that we hope mirror pulmonary metastatic disease. And if you treat the animals with vehicle, the tumors keep growing. If you treat with the HIF2 alpha inhibitor, again, you see, if anything, tumor stasis or regression. And not surprisingly, this translates into a survival benefit in a Kaplan-Meier curve. So at this point, you want to declare victory. This is what you hope to see. You write the paper. This is great. But if you're really being honest, this could all be a sleight of hand. It's plausible that this drug is acting by inhibiting HIF2 alpha. I showed you the pharmacodynamic effects. I showed you there's an apparent dependency on HIF2 alpha. But if you're really being honest, maybe this drug carries lots of other baggage. This drug also inhibits X, Y, and Z. In fact, this is actually rat poison. And this wouldn't be very interesting if what I just showed you was an off-target effect of this drug. And so for the young people, your homework assignment for tonight is to read this paper. But we've gotten really obsessed with this because so much of the medical literature just can't be reproduced by our colleagues in pharma, or at least it's been misinterpreted. Now, part of the problem is every assay I just showed you is what a screener would call a down assay: decreased transcription, decreased proliferation, decreased tumor growth, et cetera, et cetera. And screeners hate down assays for 2 reasons. First of all, the signal to noise is almost always better with an up assay. So for example, imagine this imaginary screen. Here are my signals. You immediately see the up. It's much harder to see the downs. And in fact, if the noise was high enough, you probably wouldn't even be able to see the down. So that's reason number 1. But reason number 2 is everyone knows that there are more uninteresting ways to make a complex system work less well than to make it work better. So you get mired in uninteresting positives that are simply quite trivial when you do a down assay. And so, for example, if you don't believe me, the students can go out to their automobiles later and start randomly removing parts from their internal combustion engine
and report back how often the car goes faster. Or how often have you heard of a professional athlete who got injured and then performed better, okay? So there's just too many ways to make a complex system work less well. And in cancer biology and cancer pharmacology, we've almost always set up our assays to be down assays. And this is particularly important because now you're just set up for the off-target problem where you're actually getting fooled. And so, of course, we add perturbants to cells. I just showed you a drug. It could be an siRNA, now it could be CRISPR. And we'd really like to believe that everything is on target, but if you're being intellectually honest, what you've just seen could actually be due, at least in part, to an off-target effect that you can't anticipate. So that's the bad news. The good news is real geneticists have taught us for years there are various ways that you can try to address this. So one of which is to try to discover or engineer a version of your target that's now resistant to the perturbant and see if you can reverse the phenotypes. Now, in this case, we were quite lucky because Gardner and Bruick had already identified a point mutation of HIF2 that would prevent drug binding to HIF2 And so Chin, Frosch, and my lab exploited this and engineered renocarcinoma cells using CRISPR that were either expressing wild-type HIF2 or this drug-resistant version. So for example, here's a typical soft agar assay you might see in a paper. She's treating these renocarcinoma cells expressing wild-type HIF2 with either 0.2 or 2 micromolar PT2399. She sees a decrease in soft agar growth, but how in the world do you know what that actually means? And parenthetically, I will tell you that at 20 micromolar, this drug kills every cancer cell. We've looked at even cells that don't express HIF2 So right off the bat, there is a concentration above which you can engage all sorts of alternative mechanisms. But at least this result is on target, even though no one usually does this rescue experiment. But here I can, we're pretty confident this is an on-target effect because if the cells are expressing this HIF2 they don't care anymore about the drug. And likewise, we can reverse most of those anti-tumor effects in vivo. And so here's the result I showed you earlier. Here are cells expressing an empty vector, and as I showed you earlier, they're inhibited by the drug, whereas now if the cells are expressing this drug-resistant variant, now they're largely resistant to the inhibitor. So for this and other reasons, we think these effects are on target. Now, lastly, in this paper, we committed an egregious sin, which is we actually did this experiment with 8 renal carcinoma cell lines, and 4 of them responded like I just showed you, and 4 of them, to our surprise, did not. And we made the terrible mistake of actually describing this in our paper. And the editors and reviewers said, oh, if you only knew why those other 4 didn't respond, this would be a great paper, but you don't know why. And I said, whoa. You know that what a lot of people would have done is they would have cherry-picked the responsive cell lines and then left to another day why all the cell lines didn't act the same way. And this is one of the many problems in the literature. So wouldn't it be better if we just tell people this is what we saw, and maybe some smart person, probably not in my lab, will eventually figure out why there's this heterogeneity. And guess what? Heterogeneity is what we almost invariably see in the clinic anyway, so why don't we start acknowledging it in the laboratory? So here are the data from the phase 1/2 trial with this HIF2 inhibitor. These are patients with metastatic renal carcinoma. They've been highly treated previously. The median line of prior therapy was 4. So these people had anywhere from 1 to 7 lines of therapy. They get treated in this phase 1/2 study with the HIF2 inhibitor. And remarkably, some of the patients are on study for over a year. That's the dotted line here. This is a so-called swimmer's plot, by the way. So each line is a different patient. The purple star is a complete response. The yellow stars are partial responses. The arrows are patients who are still on study, doing well. So at least some of the patients are doing quite well. Some of the patients, however, are not doing well at all. And so I think this begins to speak to this heterogeneity problem that we're only partially understanding. So for example, we've now learned in the laboratory that if you picked up a p53 mutation along the way, you don't respond to this drug, at least in preclinical models. We also know, frankly, that some of these patients were simply underdosed if we look at the pharmacokinetic data. So I showed you the rescue experiment with the drug-resistant HIF2 but I'm sure some of you are saying, well, yes, Bill, but you had delivered on a silver platter this drug-resistant version of HIF2 I'm usually not so lucky, and I usually can't find this in the Sigma catalog. What's a poor person to do in general? But fortunately, a number of solutions exist, and I'll just show you one that we found to be quite robust and we'd like to use. So another one is you take a vector for your putative target, and then you grow it in error-prone E. coli, so now the cDNA gets peppered with random mutations. This has been done by a number of people, but certainly George Daly and Charles Sawyers did this with BCR-ABL and imatinib. You take the randomly mutagenized vector, you put it into a drug-sensitive cell line, you treat with the drug, and then you sequence the plasmid DNA from the survivors and look to see in secondary assays whether you've successfully recovered drug-resistant alleles. And so just to show you an example, and in the interest of time, I'm not going to show you the data, but we identified that in VHL defective renal carcinoma cells, they overproduce various histone demethylases, and as a result, they're highly dependent on the EZH1 methyltransferase, and we had gotten a tool compound called EZ05 that inhibits both EZH1 and EZH2, and we could show that this tool compound inhibits the growth of VHL defective renal carcinoma cells, at least those that are expressing wild-type EZH1. But the question again is this on target or not? But we did the trick I just showed you a moment ago, and this was done by Abhishek Chakraborty, and he was able to, using that trick, identify a number of drug-resistant versions of EZH1 that in a test tube are resistant to the inhibitor, and in cells confer resistance to the drug. So we can say that this is at least on target also. Now, one of the things that's happening in my world is there's now a growing appreciation that there are actually a lot of these 2-oxyglutarate or alpha-ketoglutarate dependent dioxygenases. So we talked about the HIP prolyl hydroxylases. You studied in college biochemistry, perhaps, the collagen prolyl hydroxylases. It turns out that many of the histone demethylases belong to this family. The TED enzymes that are implicated in DNA demethylation and epigenetic regulation, as well as many others that frankly are orphans. Now, the reason I show you this is because we're not too far from where Otto Warburg lived, and as you know, Otto Warburg in the middle of the last century argued that altered metabolism could be a cause of cancer, and in fact he won the Nobel Prize. But subsequently, in fact for decades, there was a controversy: is the altered metabolism in cancer a cause of cancer, a consequence of cancer, both, or neither? And what he lacked was a genetic smoking gun. You know, how would you really know whether the altered metabolism he was describing was causal? And I think that has begun to change because we now know there are some families or tumors that are linked to inactivating mutations in succinate dehydrogenase or fumarate hydratase, and these lead to the accumulation of succinate and fumarate respectively, and at least in a test tube, these can inhibit various 2-oxyglutarate-dependent enzymes. In fact, you may remember that succinate was the product of the reaction, and it will feedback inhibit these enzymes. And then we learned from the work of Hai-Yan and Bert Vogelstein and others that some leukemias and brain tumors and other tumors have gain-of-function mutations in isocitrate dehydrogenase 1 or 2 that allow these mutants to now make millimolar amounts of a small organic molecule called 2-hydroxyglutarate, which again in a test tube can affect these various enzymes. And so when I was a clinician, they used to say there were two kinds of doctors. There are the lumpers and there are the splitters. So I'm sort of a lumper, so I try to put everything in one big tent. So this is the big tent view of how at least altered so-called oncometabolites can cause cancer. They do so by modulating these various enzymes. And we and others have been beginning to understand which of the enzymes are important here. So for example, in acute leukemias driven by IDH mutations, it's quite clear that TET2 is probably one of the critical targets. But we also wanted to ask another question, which is transformation by, for example, IDH mutations actually going to be reversible? Because there was a growing suspicion that these mutations would cause cancer by causing epigenetic changes, which again is probably true. But a lot of people then made the next leap and said, if it's epigenetic, it will be too slow to unwind, and there won't be a therapeutic opportunity. So Julie Lossman in the lab realized she needed cellular models where IDH mutations would score, or mutants would score as oncogenes, and she could then use those models to address mechanistic questions, but also therapeutic questions. And so she stumbled upon a leukemia cell line called TF1 cells that, despite being leukemic, require cytokines for proliferation in vitro. So she then introduced into these cells this canonical R132H IDH1 mutant compared to wild-type IDH1 or an empty vector, and she saw that this canonical IDH1 mutant conferred cytokine independence, So that was good, and not shown here, it also blocks differentiation, so that's also good. So at least in this system, mutant IDH is scoring as an oncogene. And then Julie decided to swing for the fences. And another piece of advice for the students is, be bold, don't be cowards. So Julie decided, if it's really about 2-HG, I should be able to transform these cells just giving them 2-HG. And there was a little trick involved. She collaborated with Ryan Looper, who produced an esterified version of 2-HG that gets into the cells and then gets trapped upon deesterification. So here she's treating the cells with increasing amounts of 2-HG, and it turns out indeed 2-HG alone is sufficient to confer cytokine independence. And there's one more trick here, and that is a geneticist's best friend is a point mutant, and a chemist's best friend, or one of the chemist's best friends, is an enantiomer. So it turns out for 2-HG there's an R enantiomer and an S enantiomer. So here's the tumor-relevant form, the R form. Here's the same experiment with S2-HG. And even though S2-HG gets into the cells as well as R2-HG, only R2-HG can transform these cells. And again, in work I'm not going to show you today, our work and others suggest that the relevant target of R2-HG here is TET2. So now the question is this reversible? So we obtained a tool compound developed by Agios that binds to this mutant IDH, this IDH1 R132H, and it prevents it from producing 2-HG, shown in this very busy graph over here. So now Julie, having a cell-based system, she can now ask whether the inhibitor will also suppress proliferation. So let's first take her isogenic cells expressing wild-type IDH1, or this canonical tumor-associated IDH1 mutant, and let's treat with DMSO or the inhibitor, and let's measure intracellular 2-HG. Right? So as advertised, this drug really has the intended pharmacodynamic effect. It lowers, but I will point out doesn't normalize 2-HG levels. And now she can take those cells I showed you earlier expressing the mutant IDH1, but now treat them with the inhibitor, and again, you see a decrease in proliferation. But again, for the students, the most dangerous result in all of science is the one you hoped you would see. Okay, so this is the one where you go running down the hallway, say, look, it really worked, right? This compound really does what we had hoped it would do. We can really block the proliferation of these cells with a IDH inhibitor. But if you're really being honest, this could be true, and unrelated. Again, maybe this is rat poison, and maybe it does lower 2-HG production, and maybe it does inhibit proliferation, but it's true, and unrelated. An even worse interpretation is, this is such a toxic, noxious chemical that the cells are really sick, and because they're sick, they're not proliferating, and because they're really sick, they're not making a lot of 2-HG. That would be a really trivial, uninteresting explanation, and yet I suspect that is the explanation for half of the cancer pharmacology literature. So Julie, fortunately, realized that if she's on target here, and the drug is really acting by blocking 2-HG production, that if she treats cells where she's already giving the cells the 2-HG, they should be completely insensitive to the inhibitor, and that's exactly what she saw. And not shown here is the fact that if she gives the esterified 2-HG to these cells, they start growing again. So it really does look like it's about 2-HG. Fortunately, this helped galvanize efforts to bring these drugs into the clinic. For technical reasons, the mutant IDH2 inhibitor went into the clinic first, but now the mutant IDH1 inhibitor is also in the clinic. But here are phase 1 data from patients with acute leukemia linked to IDH2 mutations, treated with this first-generation inhibitor from Agios. And quite remarkably, even in the phase 1 study, some of the patients went into complete remission and some went on to have consolidated transplants. Now, with larger numbers, I think the response rate is closer to 30 to 40%. So once again, heterogeneity rears its ugly head, but at least it's a foot in the door. Okay, so now I'm going to shift gears. So if you didn't find that interesting, it's done. Okay, so we're going to move to something new. Okay. So now I'm going to tell you about the story of thalidomide, and as you probably can tell, I think we, like all of you, like medical detective stories. And you may know that this drug was available over the counter in Europe in the 50s and 60s for the treatment of insomnia and morning sickness, which was particularly cruel and disastrous because if you were a pregnant mom and you took this drug, there was a chance your children were going to have these horrific limb defects that led to the term thalidomide babies. And I can remember as a child seeing thalidomide babies. The drug was largely kept out of the US market, but you would still occasionally see, growing up in the 60s, thalidomide babies who had no arms or or no legs. So now you have this infamous, terrible drug that's now been taken off the market, but then through serendipity, thalidomide-like drugs, including lenalidomide, were found to be extremely active in the treatment of multiple myeloma, which is a B-cell malignancy. So now you have this terrible drug, but it's a wonder drug for multiple myeloma, and we don't even know the mechanism of action. So the foot in the door came from Hiroshi Handa, who immobilized thalidomide on a cephros bead and looked for proteins that would bind, and he identified the cereblon ubiquitin ligase. And this was interesting to us because architecturally, this ubiquitin ligase loosely resembles the VHL ubiquitin ligase, and here it's the cereblon protein that's going to be the substrate adapter. And in their final figure, which was probably prompted by a reviewer putting a gun to their head, they then went on to show that at least in a test tube, thalidomide could inhibit the auto-ubiquitination activity of this complex. And so the simple view in 2010 was the IMiDs are cereblon antagonists. Fortunately, however, if you then looked in the clinic and studied patients with multiple myeloma who had responded to an IMiD and then became resistant, you got a surprise. And the big surprise was that not infrequently when patients became resistant to these drugs, their myeloma cells were no longer producing cereblon. Conversely, the higher the levels of cereblon, the more likely you were to respond to the drug. Now, especially this first point is a head scratcher. I mean, if you're killing the myeloma cells by inactivating cereblon, it's not clear why getting rid of cereblon would make you resistant to the drug. And so, interestingly, the people who were writing these papers said this provides genetic evidence that the target is cereblon, but no one was willing to then make the next logical leap, which is, and the drugs are not antagonists. And likely what's happening is cereblon, once bound to a thalidomide-like drug, is acquiring the ability to destroy some myeloma oncoprotein. So to look for this oncoprotein, Gong Lu in my lab made a bicistronic vector modeled in part on earlier work done by Steve Elledge. But in Gong's vector, your protein of interest is fused to firefly luciferase, and then the same transcript encodes renilla luciferase. And then to compare apples to apples, we have two irises here, so they're both under iris control. And now the idea was you treat with a perturbant, and now you look for changes in the ratio of the two bioluminescent reporters to infer changes in the stability of the protein of interest. And then as sort of a tour de force, Gong made a library of about 12,000 of these with the help of Mark Vidal, so we have 12,000 different proteins of interest. Gong then arrayed them in 96-well plates with the help of our friends at the Belper Center, and then he treated the plates with either lenalidomide or vehicle, and he looked for proteins that either went up or down in stability as determined by changes in the ratio of firefly to renilla luciferase, as shown here. So since this work is published, I'll just tell you that the two true positives from this screen were IKZF1 and IKZF3, and I should point out the same conclusion was reached by Ben Ebert working down the street from me. And so here you're showing in secondary assays that if you treat cells expressing the IKZF1 or IKZF3 luciferase fusions, and you treat the cells with lenalidomide, shown in red, that you see a decrease compared to the vehicle-treated cells. And this is specific because here are 3 paralogs, and they're not affected. And this wasn't an artifact of being fused to firefly luciferase, because if you replace the firefly luciferase with an HA tag, you still see downregulation. And further, if you treat real myeloma cells, such as these 2 cell lines here, with lenalidomide, which again is a slightly more potent version of thalidomide, you see downregulation of IKZF1 and IKZF3. And I should point out IKZF1 has multiple isoforms. And this is specific too because there is no downregulation if you co-treat with a drug that blocks netylation. And when you block netylation, you block the activity of ubiquitin ligases, such as the cereblon ubiquitin ligase and the VHL ubiquitin ligase that have a colon. So that all look very good. Now in biochemical work, I'm not going to show you, it appeared that the drug, once bound to cereblon, was either going to be on the substrate docking site or was going to be inducing an allosteric change in cereblon. And the beautiful work of Eric Fisher and Nicholas Thoma showed us it was the former. So the IMiDs are like a piece of glue or gum on the putative substrate docking site of cereblon. And this leads to the recruitment of IKZF1 and IKZF3. But again, this is just true, and unrelated, at least with respect to the anti-myeloma activity of these compounds. And so the first thing Gang did was he identified 3 effective hairpins against IKZF1 and 3 effective hairpins against IKZF3. For reasons we don't completely understand, if you downregulate IKZF1, you also downregulate IKZF3 and vice versa, and IR4 turns out to be a downstream target gene. But what's very clear is if you do fitness assays in myeloma cells compared to cells with negative control hairpins, the cells that got these hairpins drop out over time. Okay, but I hope the students have appreciated that I really hate down assays, and this is just another down assay. Okay, so you knock out some really important gene, and the cells don't like it. Are we supposed to declare a victory? Are we supposed to, call the editor of Cell? I don't think so. This is not a very interesting result. So we need an up assay. So the up assay was as follows. So I showed you these data a moment ago. So Gang actually was smart enough to realize there are only 7 amino acid differences between IKZF1 and IKZF2. So he mutated them one at a time until he arrived at the critical difference. And using that information, he can now make IKZF1 and IKZF3 point mutants that are resistant to the IMiD, and then ask if that rescues or reverses the sensitivity to an IMiD, and that was what he saw. So here's the drug-resistant IKZF1, and here's the drug-resistant IKZF3, and a competition assay conferring resistance to an IMiD. Okay, with that, I'll thank you very much for your attention and be happy to take any questions.
Very nicely talked about the pitfalls of negative assays and the need to use rescues as a way of showing specificity. We had a very nice example of actually turning a negative assay into a positive assay in the case where it's involving protein degradation. So I was wondering in the cancer field whether you or others are generally moving towards positive assays for screening.
Well, it's certainly why I wrote the review article I did, which was in response to a number of articles that have appeared over the past several years from a number of our colleagues in pharma telling us that 50 to 90% of the time, the results that are being published with respect to target validation are either not true or they're not robust. And by the way, I had this argument with some of the people in my lab. They think robust means big, great, wonderful, whereas robust really has a specific meaning. And so, it's supposed to mean it's true under a variety of conditions and can withstand perturbation. So I point out if the results, true at pH 7.2, but it's not true at pH 7.22 or 7.18, and it's only true on Tuesday and it's not true on Wednesday, you start to worry a little bit that this is not going to be robust. So they tell us that the things can't be reproduced, they're not true, or they're not robust, or they've simply been wildly misinterpreted. I mean, the data are the data, but the overriding conclusion that this is a good target for this cancer is simply not supported. So I think part of it is doing more rigorous genetics and trying to get away from the down assays. But another thing I wrote about in this recent editorial, and the word I didn't use today, is an old-fashioned word. It's called corroboration, right? So if you have 3 or 4 lines of evidence that something is true, it's more likely to be true, of course, than if you have 1 line of evidence. So I don't want to suggest that you need to do a rescue for every single perturbant you use, but it would be nice if maybe you did a rescue, for one of your experiments, and then you had corroborating lines of evidence. For example, my pharmacology colleagues point out, there are some drugs that have been around for a long time. They're very well studied with respect to specificity and potency. So I don't think you have to invent the wheel every time. Having said that, my colleague Paul Workman and my friends in pharma will also agree. Another trap academics fall into is they want a tool compound. So they go to Google or they go to PubMed, okay? And it turns out that the number of citations usually reflects in part how old the drug is. And the older the drug is, the less specific and the less potent it is. So real pharmaceutical people know that, oh no, you don't want to use that thing from the Cal Biochem catalog. We never use that. You want to use the thing that's come out in the last 6 to 12 months. So I would say even with chemicals, you got to be a little careful. The Broad Institute in Boston recently bought 500 chemicals from commercial vendors trying to make a little chemical library, and then did quality control on the chemicals they just bought. And in 100 of the 500, there was either no active ingredient or it was a different active ingredient than was on the label. Now, I have to say, as a molecular biologist, I found this shocking None of my chemical colleagues found this shocking. So, if someone gave me a plasmid when I was a young person, I assumed it was wrong until proven otherwise. Now, Egon and I did restriction maps. I think now you just send it and get it sequenced. But you get a chemical, you just say, well, that's what it is. It's on the label. It's got to be that chemical. It turns out that's not a good assumption always.
The two forms of the two enantiomers formed under all similar conditions or different conditions?
Yeah. So it turns out people much smarter than me seem to understand why the mutant IDH can only produce the R form and not the S form. And it was originally thought this was a neomorph, that the enzyme never made R2HG. Now it turns out under certain conditions there's a teeny bit of R2HG being produced. But to your point, there are other enzymes in the cell that under certain conditions can make S2HG. So for example, Craig Thompson has shown that under profound hypoxia, lactate dehydrogenase makes S2HG. So they're both present. And in fact, we have enzymes in our cells that remarkably can clear these two different enantiomers, and there are even rare genetic syndromes linked to loss of these dehydrogenases.
But there's a reverse reaction, so that it can be formed back, no?
That I don't know. I haven't seen that. I haven't seen that reported. There is a dehydrogenase that will take it away. I haven't seen anyone. It should be possible, but I haven't seen that.
Yeah. How many substrates are being recruited to cereblon by thalidomide? And how specific is that? And is it overlapping sets of substrates that are responsible for the different effects of coma treatment and treatment of insomnia and the severe development effects?
Okay. So it's a great question, and it's one of the reasons why Celgene is the company that owns these IMiDs. And it's one of the reasons why when they saw my postdoc as the first author on that paper, they hired him. So he now works for Celgene. And talk about having money fall out of the sky, because they bought the IMiDs for virtually nothing, because who wants this drug? And then, as I said, through dumb luck, it was found to be an anti-myeloma drug. And so then they had their chemists do what chemists do, which is to make all of these variations and just empirically decide which ones become more potent as anti-myeloma drugs. And so, for example, they went from thalidomide to lenalidomide to pomalidomide, and then they have all sorts of composition of matter IP around every permutation. So now to your question, it's becoming very clear that what they have is a treasure trove, because you can start making chemical modifications of the IMiD and you start recruiting different substrates. And so, for example, one that's been published by Ben Ebert and others is, I think it's casein kinase 1 alpha, I might be wrong, but it turns out this is a gene that's deleted in these 5q minus myelodysplastic syndromes. And so instead of having 2 copies of the gene, they have 1 copy of the gene. So they're already sort of teetering at a 50% level, and they're hypersensitive to some of the IMiDs. And as I said, it's very clear in proteomic and other experiments that these different IMiDs are able to latch on to different proteins. And we're just starting to learn some of the rules. And then you may know that Jay Bradner's group and Craig Crew's group, and I think the first author of one of those papers is now in Vienna, Greg Winter, showed that if you're smart enough, you can actually take the cereblon binding portion of the IMiD, fuse it to a molecular hook that you already know will bind to your protein, and if you're lucky, get cereblon to now degrade your protein. So one of the companies that was on my disclosure slide, I think, is possibly moving in that direction of trying to design molecular glues based on cereblon. And then more recently, Deepak Nishawan had a lovely paper in Science where he's found another example of a drug that recruits a different ubiquitin ligase to a different substrate.
Last question from Anna.
Very nice talk. I was wondering if you could give us some more insights on the resistance mechanism to the HIF-2α inhibitors that you mentioned.
Yes. So the one that we understand best is we found that some of the cell lines that are resistant have acquired a p53 mutation. So that's good news and bad news with respect to kidney cancer. In kidney cancer, p53 mutations are quite rare if you look at, for example, the tumors that were initially removed by the surgeon. But if you start to get into metastatic disease, and you start to get into metastatic disease after multiple lines of therapy, you start to see the p53 mutation prevalence go up. And so I'm hopeful where we're going to get is that the patients will have their p53 status reassessed. And ideally that would be done by looking at circulating DNA to kind of survey all of the metastatic disease. And I think if they have P53 mutations, they may not be good candidates for this drug.
Okay, let's thank Bill again.